And again, welcome to this presentation, Optimizing Revegetation of Highly Disturbed Soil on Mine Sites and Construction Sites Through Beneficial Soil Health Management Practices, presented by Paul Storer from EnviroStraw. Welcome, Paul, and I'll turn the microphone over to you.
Thanks very much, Kim.
Yeah, I gotta watch this. Sorry, guys.
Is everyone ready?
Yep.
Okay, I think we need to turn off our audios somewhere. Okay, thank you very much, Kim. What I'm going to do is present a talk on soil health being a key to success in rehabilitation strategies, and go through some of the results of what we've been doing in the field here in Australia. Now, the focus of the talk is basically looking and highlighting the environmental benefits of using soil microbiology and regenerative and revegetative practices for the remediation of highly degraded, highly depleted, and highly disturbed soil. Part one of the talk will look at defining soil health and getting a basic understanding of the key functions of the soil. So we'll look inside the soil as a black box and see what really makes the soil tick. And then more importantly, touch on how we can turn on the mechanisms of the soil in part two. And this is where we'll look at using EnviroStraw's Enviromatrix Ecm, which is a biotic growth media, how it triggers and optimally feeds the beneficial microbes. And this is a typical example of EnviroStraw's biotic technology that we use when we're implementing hydraulically sustainable revegetation programs. So what is soil health? Firstly, what we need to do is look at the functions of the soil, what it actually does. Soil is the habitat for diverse micro and macroorganisms. It stores, decomposes and recycles organic matter, organic carbon and nutrients. It stores and filters water. It modifies the atmosphere, either through emissions or sequestration. And ultimately, it's medium to sustain plant growth. If we take into account these main functions, we can then start to develop a definition of what soil health is. And the main definition we're going to be using today is the capacity of the soil to efficiently function as a living dynamic organism that responds either positively or negatively to the way it is managed and maintained. So being able to define and understand soil health really matters. This helps us adopt best management practices that can positively influence self-sustainability. And this is very important in revegetation, as I mentioned, particularly in a drying climate. So if we look at the mechanisms that are going on within the soil, inside the black box, so to speak, we can start off with dirt. Typically after a construction site or closing of a mine site, we have a lot of dirt there, which is basically depleted in beneficial microbes, depleted in essential mineral nutrients. And what we need to do is look at how we can turn the soil back on, what makes it to make it into a healthy living soil. First thing that we need is mineral nutrients, and we can supply those using our technology where we've developed a controlled release fertilizer technology using natural mineral ores that become bioavailable through microbiology. The second thing that soil needs is some beneficial microorganisms. Now, after we've done a lot of construction or we've left topsoil standing in a huge pile for a year or so, a lot of the beneficial microorganisms are depleted, or the balance of them has changed. And so if we do have microorganisms, they generally are bacterially dominated. For most of our plant systems, we need a fungally dominated system. So we have to put those beneficial microorganisms back in. And so we've developed a multi-strain inoculum where we can put essential starter microbes back into the system, particularly bacteria and fungi, that will drive this system. So these beneficial microbes are the drivers of the mechanism of the soil. We then need water, whether it be irrigated water or from rainfall. They then activate the microbes and they eat the insoluble mineral nutrients, and we start to turn the mechanism on. And so slowly the soil switches back on. Once this happens, we start to see organic carbon being laid down, firstly through root systems that are being grown within the soil, and also through a thing called microbial biomass carbon. And I'll talk about that in some detail. These beneficial microbes that we inoculate into the system grow organic carbon in the soil. Now, the importance of organic carbon, which is highly depleted in disturbed soils, is that they act as a water sponge. They hold moisture in the soil that is then plant available. We also use biocompatible inputs, because once we've turned this mechanism on, we've got these beautiful microbes working, we don't want to then go and put in chemicals or things that are going to either damage or negatively impact on the beneficial microbes and then wind the soil mechanism backwards. So if the take-home message from this whole talk is that soil should be alive, it should be a dynamic living organism powered by beneficial microorganisms. Once we have that happening, we then get more efficient use of water and nutrients, which results in improved soil health, improved vegetative cover, bigger root systems, more microbiology in the soil, increased soil carbon. This helps with reduced erosion, and ultimately we're looking at economically and environmentally positive outcomes. Through all of this, it's all about risk mitigation, whether it be from perturbations in the climate to a lack of rainfall to damage to soil and infrastructure of the soil. So again, looking inside the black box, we can see the power of the microbes. Technology now, DNA technology, microscopy and so on, we can actually start to look at living systems within the soil and how these microbes can build this microbial biomass carbon, and also how they can be involved in biological soil stabilization. If we take a look at a conventional system, we have a plant with a root. We apply water-soluble fertilizers, and they basically wash past the roots, and some of them are taken up, some of the nutrients are taken up, approximately 30% at best, because what happens is a nutrient depletion zone around a root, which is about 1 millimeter to 1 centimeter out from the root system, where the plant can pull nutrients in. If it's outside of that region, then the nutrients aren't available to that plant. If we look at the biological system that we use in EnviroStraw, we use a whole load of microbiology, starting with mycorrhizal fungi, and they grow out into the system, attach to the root and grow out into the soil. And as you can see, huge volume compared to the conventional system. So they can pull nutrients and water from a long distance away. And effectively, we can increase the drawing power of this root system up to 1,000-fold. In addition, we've got beneficial bacteria and fungi that are free living, that move in and out, pulling nutrients in as the plant requires them. And so, as I mentioned before, we've developed a control release fertilizer from natural ores where all the macro and micronutrients are in the system. And these are non-water soluble, so they'll sit within the profile and not leach away. The microbes will come and pull the nutrients out as the plant requires them. it's not required, then they'll stay in the soil and increase the soil fertility for subsequent years. So what we're starting to see using this biological system is a high nutrient use efficiency. We don't have to put too much nutrient in to get effective uptake by the plants through this microbial system. Whereas over here in the conventional system, it relies on its root system, doesn't rely on beneficial microbes. So we might get a few microbes, particularly bacterial dominance in the root, but they don't start to spread out into this much wider area and into this laid down organic carbon. Again, through the microscope, we have a root system on the left-hand side and a mycorrhizal fungi growing out. This is after 2 hours, and there's a hyphae, which is like a hose that pulls nutrients and water into the plant. Wrapped around it, we've developed a biological bacterial technology that is, in fact, an envelope that protects that hyphae from movement in the soil, from toxins in the soil, and so on, and creates a non-hostile environment. Now, you can actually see the bacteria moving around here, and this envelope is many-fold larger than the actual hyphae. And the beauty is that this is fixed carbon and other nutrients that the bacteria bring. The mycorrhiza release glomalin and other nutrients, and they bind into this. And we have a symbiotic system where the bacteria and the fungi work together and form these biofilms within the soil. And this is basically carbon concrete or sequestered carbon. More importantly, when we're using this technology, we're starting to increase the microbial biomass carbon. And this confocal microscopy shot video will show, this is sped up 50 times, but it basically shows how the mycorrhiza spread, and they form these networks in the system. And you can see them spreading, forming networks, and there's the bacteria and the fungi growing out into the environment. And so we start to get this network, and you can start to see a sponge effect occurring. So these networks form these bacterial and fungal lines, and between them are air spaces. And these pockets can fill with water when it rains or it's irrigated, so they can hold on to moisture, and then they can pull it back when things dry out. So this is key to what we're doing in the biological system, encouraging the beneficial mycorrhizal hyphae and the beneficial bacteria to form this carbon sponge within the soil. Within there are all sorts of things like biopolymers, biofilms from glomalin, as I mentioned, the carbon concrete within the soil. And these help to form soil aggregates. They help with holding moisture and water stability within the soil profile, which also help increase the shear strength and decrease soil erosion. And so you've got big root systems holding on to the soil, as well as these networks that are bacterially of bacterial origin and fungal origin, and it turns the soil into a water holding sponge. And research has shown that for every 1% increase in organic carbon within the soil, you can hold on to approximately 144,000 litres per hectare of water. So that's an equivalent to, say, 14 millimetres of rainfall. And in a drying climate and a drying environment, this can be the critical thing between plants surviving, particularly in the early germination stages of the life of the plant, through to the longer term fixation of carbon through sequestration. In addition, these mycorrhizal hyphae help in forming these microbial aggregates within the soil, so binding particles together, and so we start to get pore spaces forming, structural tilth within the soil. And this is very important for dispersive or structurally unstable soils. So that's just given you a brief introduction into soil health, soil function, and so on. How do we go about actually improving the system, particularly when we're looking at depleted or degraded soils, post-construction, post-mining, and so on? Well, we've developed a product called Enviromatrix Ecm, which is a hydraulically applied biotic growth media. It's a one bag product, so everything's included in there: the fertilizer, the microbes, the straw, the binders, and so on. And this is designed to enhance soil health by firstly improving the fertility, increasing the microbial activity, and ultimately leading to increased soil carbon. And I'll just mention in passing that this product won the IECA 2020 ICE Award for excellence in the environment innovation. It's basically a cutting-edge technology that we've developed over the last 10 years, but more specifically, this product we've been working on for a couple of years and now rolling out. It's been validated in the field, and we've done an extensive amount of lab testing and research into it. So as I said, basically it's a simple to use one bag product for erosion control, and it's a biotic growth media. All you have to do is add seed and water, and away you go. The fibers in there are mechanically and thermally processed, and we use biocompatible tackifiers and binders. It includes a multi-strain suite of purpose-selected beneficial microbes, and ultimately it has the control release fertilizer, so it's not going to leach away into the environment. Provides a viable economic and environmental advantage to our system. And ultimately, this is all about getting successful outcomes, and this product is one of those that we've had amazing success with. So what does Enviromatrix do? It's basically a high-performance biotic growth media, builds fertile soils. It looks at improving soil nutrition, soil health, and soil quality. The soil nutrition is through these control release fertilizers that are incorporated in the mix, and this can be hydraulically sprayed out with the mulch, so it's all in one product. Included is a multi-strain microbial technology, a suite of about 22 beneficial microbes that we've specially selected to go into really hostile environments. And then we look at the soil conditioners and amendment technology, and we only use biocompatible ones that are not going to affect our microbiology. All of our programs are based on comprehensive soil tests, and then we look at using those to work out the application rates and look at the climatic conditions. We look at what's happening with the slopes that we're applying this stuff to, and so on. And we don't get caught up in pH. Ultimately, we're looking at growing large root systems. So it's everything's happening below ground in that black box. And included in that, we have that increase in microbial biomass carbon. So here is your water sponge below ground, and there's the plant above ground. This leads to microbiological soil stabilization. And so we look at balancing the chemical and physical systems as well as the biological parts of the soil. And this leads to increased soil health, soil quality, and productivity. Ultimately, we get improved nutrient use efficiency and water use efficiency, which leads to this environmental and self-sustainability. So we've done a huge amount of research in the real world, looking at comparison between the Enviromatrix compared to inorganic fertilizer, Hydromulch programs. We've done a lot of university tests and independent tests looking at things from overland flow, rainfall simulation, how big the root systems are, what the microbiology is, how much organic carbon is built up. the water holding capacity and water use efficiency of the Enviromatrix system. And ultimately, it comes down to the positive changes in microbiological systems within the soil. And without going into too much detail, we're seeing basically that over time, we're going from a bacterial dominance to a fungal dominance. We're increasing specific recycling microbiology, and this is helping in nutrient uptake. So we're finding that we're getting better nutrients, for instance, increased organic carbon in the soil. We're getting improved nitrogen fixation going on with our free-living nitrogen fixers, and then nutrients like phosphorus and that are being pulled up fairly efficiently. And we've done a whole load of case studies that are available on request, and you can contact us either through the website or personally through email, and we can pass those details on to you at the end. I will end at that point.
Great, Paul. We've got just a couple of questions. Is EnviroStraw product a dry product? What is the moisture content in the material?
Yes, so basically it comes as a dry product. The straw is dried down, and so the biological fertilizer, the microbes are in a stabilized form. So we've developed polymer technology to hold the microbes in stasis, and they only activate once the plants germinate, when there's enough water around. So yeah, it's comes in a bag.
And we had someone ask for the cost of the material, but I'll pass their contact info off to you.
Yes.
Another question. This is from Mark from Profile. Great to see you, Paul. You mentioned that Enviromatrix is an all-in-one product. Do you have the testing data to support its efficiency under simulated rainfall conditions, such as those in the prestigious landlocked testing facility? How effective is the material at controlling erosion?
Excellent question, and hello, Mark. Yes, we've done a lot of work with landlock, and I'll quickly bring this up. I don't know if you can see that. But yes, we've done overland flow, we've done rain simulation testing with landlock, and it's a beautiful system. Because of the fibers that we use, we get a very good matrix forming on the surface. They bind to the soil profile very well. We then have the microbes forming the interlocking carbon chains down into the soil and biofilms. But yes, to answer your question, the testing has been done through landlock in Australia.
Great. One more question from Tom down in Denver. What is the typical application rate?
So depending on obviously the climatic conditions, the soil profile and so on. So we're looking at between 5 tonnes per hectare to 10 tonnes, depending on the profile. And again, we use the soil testing, soil analysis to work out what nutrients are required within the system, and then we're applied at specific rates. So typical to most other hydromulch systems.
Okay, just a couple more questions. Do you need to apply extra water soluble fertilizers to the program?
With the Enviromatrix, all of the nutrients are built into the one bag. You don't need to apply any other fertilizer, particularly water solubles. We stay away from those. Basically, the nutrients, the fertilizer that we have within the system are designed to feed the microbes, and then the microbes feed the plants. So we don't need to apply water soluble because most of that's going to end up down the bottom of the hill anyway.
Last one, when using the program, do you need to add extra biostimulants to the hydromulch mix to feed the microbes?
No, everything, all the food and everything is contained in bags, so we don't need to add extra humates or other biostimulants, seaweeds and so on, extracts. Everything is contained within a bag for this to develop into a self-sustaining system. And because we've got such a suite of different microbes, for instance, we've got free living nitrogen fixers in there that fix nitrogen. So we don't need to put huge amounts of nitrogen or phosphorus out because it's all contained within the bag and then within the growing system.
Great. Well, thank you for your participation today, and thank you, Paul and EnviroStraw, for all of your support. To continue this conversation with Paul, head back to the discussion board with this presentation, and don't forget to stop by and check out EnviroStraw in the expo. Thanks so much. Have a great one.
Thanks very much, Kim.
See you. Bye.
Bye now.